And today we are looking at pharmacodynamics. This is in the continuation of the lesson where we talked about pharmacokinetics and the parenteral route of drug administration. So today we are looking at pharmacodynamics. Okay, so we previously looked at pharmacokinetics and the steps there that are involved in pharmacokinetics. More here, like just a preview of what we looked at. We looked at drug absorption, okay, under pharmacokinetics. So the effect of the body on the drug. So there is absorption, there is distribution, there is metabolism, and there is excretion. So under drug absorption, we talked of transport of the drugs, that is from the gastrointestinal tract. So we established that mostly drugs get absorbed by the mechanism of passive diffusion. So in a passive diffusion, it refers to the passage of the drug molecules by diffusing from that is unionized molecules through the lipid membrane. So the drug here, what is important to note is that it is moving from a region of higher concentration to one of lower concentration. Then when you talk of active transport, we are necessarily looking at the movement of the drug against a concentration gradient. So when they are saying against a concentration gradient, that is from region of low drug concentration to that of higher, which is necessarily the opposite of passive transport because it is from higher concentration to lower. Then in active transport, it is from low to higher concentration. Okay, then now we talked about endocytosis, which was talking about invagination of the cell. Okay, invagination of the cell in order for it to take a drug a molecule which is big so that it can enter the vacuum of the cell and then it is used by the body. Then exocytosis, where we said it is the reverse of endocytosis, okay, and it is used by cells to secrete many substances that are of similar vascular process. Then we have looked under pharmacokinetic drug distribution, of which we have said this is dependent on the blood flow, and then it is also dependent on the binding of the drug to plasma protein. And the mostly transporter, the taxi that transports, the bus that transports is usually albumin. Then the apparent volume of distribution and its formula is equal to dose administered, where we divide with plasma concentration of the drug. So the volume of distribution here is very important to know how much of the drug is within the plasma and how much of the drug has been left to work on the tissue, because that one which has been bound to the protein remains inactive. Then we looked at drug metabolism. Drug metabolism, this is a reaction of drug metabolism. It is in two phases, where there is phase one. Phase one, mostly it's about the conversion reaction of the lipophilic molecules into those polar molecules by exposing a polar function of the group so that the drug can easily be excreted. So phase one involves oxidation, reduction, and hydrolysis. Then if there are some metabolites that are still lipophilic, to expose the polar parts, we need the process of conjugation. Okay, and the endogenous substrates that are important here are sulfuric acid, acetic acid, or amino acids. So these result in exposing the polar component so that they become water-soluble compounds and excreted by the kidney. Then we talked about drug elimination. The drug being taken away from the body. So drug elimination was composing kidneys. Okay, so chiefly the kidney are involved in the elimination of the drug. Okay, so also the liver, as it conjugates the drug to also release the drug through bowel and comes out through feces. Then the GIT is also involved in the excretion of the drug. Okay, so we also gave some examples under that. Then the lungs, mostly when you talk of anesthetic drugs, okay, they can be excreted as during the expiration. So, especially anesthetic drugs. Then other routes of drug uh, elimination, drugs can be eliminated in sweat, tears, breast milk, and salivary secretion. Now, this lesson we are looking at pharmacodynamics. Okay, so we are looking at it now, the effect of the drug on the body, not the effect of the, not the other way when we're looking at pharmacokinetics. So now we're looking at the effect of the drug on the body, but not the body on the drug, which is pharmacokinetics. But we are now in pharmacodynamics, appreciating the effect of the drug on the body. So most drugs exert their effects both beneficial and harmful by interacting with receptors that are specialized target macromolecule present on the cell surface or intracellularly. So receptors bind drugs and initiate events leading to alteration in the biochemical or biophysical activity of the cell and consequently the function of an organ. So drugs may interact with receptors in many different ways. Drugs may bind to enzyme, nucleic acids, or membrane receptors. So we are now looking at the interaction effect of the drug with the body, looking at the effect of the drug on the body. So drugs, as we take them, they may bind to enzymes, nucleic acids, like from nucleus, nucleic acids, or membrane receptors. So what are the major receptor families that we have in our body? So, pharmacology defines receptors as any biological molecule to which a drug binds and produces a measurable response. Thus, enzymes and structural proteins can be considered to be pharmacological receptors. So we have ligand or ligand-gated ion channel receptors. We have G protein coupled receptors, and we have enzyme linked receptors, and then we have intracellular receptors. So these are types of receptors that we have: ligand-gated ion channel receptors, G protein coupled receptors, enzyme linked receptors, and intracellular receptors. Let us now look in more details at what ligand-gated ion channel receptors are. So these are responsible for regulation of the flow of ion across cell membrane. The activity of these channel is regulated by the binding of a ligand to the channel. So nicotinic receptors, okay, and the gamma aminobutyric acid, abbreviated as GABA receptors, are important examples of light-gated ion receptors. So when we talk about light-gated ion receptors, we are looking at receptors responsible for the regulation of the flow of ions across the cell membrane. So these activities of these channels are regulated by the ability to bind to the ligands, okay, channel. So nicotinic receptors and GABA receptors are important examples under ligand-gated ion channel receptors. Now, Apart from these, we have other receptors which are G protein coupled receptors. So a second family of receptor consists of the G protein coupled receptor. These receptors contain a single peptide. These receptors are linked to a G protein having three subunits, that is alpha, beta, and gamma subunits. Binding of the appropriate ligand to the extracellular region of the receptor activates the G proteins. So, to do with the G protein coupled receptors, okay, we are saying the tissue receptors, they contain a, a single peptide, and these receptors are linked to the G protein that has three subunits, which is alpha, beta, and gamma subunits. Okay, so the receptor, these activates what we call the G proteins. Then the other type of receptor is the enzyme linked receptor. So this is a third major family of receptor that consists of those having cytosolic enzyme activity as an integral component of their structure or function. Binding of the ligand activities or inhibit this cytosolic enzyme activity. So enzyme linked receptors, we are saying these, okay, receptors consist of those having cytosolic enzyme activity as an integral component of their structure of function. So the binding of a ligand activates or inhibits this cytosolic enzyme activity. Okay, so now the last receptor that we're going to talk about in pharmacodynamics is intracellular receptors. So intracellular receptors are called intracellular receptors because these receptors are either in the cytoplasm or they are in the nucleolus. And this gives response by increasing the gene transcription. So we have the intracellular receptor. Okay, so these receptors, they can be found where? In the cytoplasm or in the nucleus, and give response by increasing the gene transcription. Okay, so that is our lesson on pharmacodynamics. So in pharmacodynamics, we are just appreciating the different receptors. So receptors, these are sites of action for drugs. Okay, so we are just appreciating the sites of action for the drugs, and we have just discussed four sites of action, okay, which is Ligand-Gated ion channel receptors, G protein coupled receptors, enzyme linked receptors, and intracellular receptors. So thank you very much for listening, and please keep studying.